DNA Depot welcomes you to How Does a Doctor Test for Lyme Disease? Kit DD231. The objective of this experiment is to learn the basic concepts related to Lyme disease, the immune system, and the ELISA test. The ELISA test is an initial screening test for the diagnosis of certain diseases. This experiment is a simulation of an ELISA test to detect circulating antibodies in a hypothetical patient's serum. Your kit will contain the following reagents: Lyme antigen, negative control, positive control, donor 1 and donor 2, secondary antibody, and substrate. In addition, microtiter wells will be found, as well as micropipettes. From your home, you will need a small cup with distilled water, a pencil or pen, and a timer or clock. In step 1, carefully label the bottom of the wells as shown in the video clip or as shown in the instructions manual found within your kit. As long as you remain consistent, either method is correct. Carefully place 2 drops of Lyme antigen into all 8 wells of the microtiter strip that you marked in step 1. Be sure you place any unused antigen back into the container for further testing. Incubate the plate for 5 minutes at room temperature while you rinse the pipette in your water container. Move water in and out of the bulb to clean it completely. Place 2 drops of the negative control into the wells labeled 1. In addition, place 2 drops of the positive control into wells labeled 2. Be sure that you rinse the pipette several times in your water container before moving on to the wells labeled 2. Continue by adding donor sample 1 to wells labeled 3 and donor sample 2 to wells labeled 4. Again, be sure you rinse the pipette thoroughly between moving wells. Incubate the samples for 5 minutes and discard the pipette used in this step. With a new pipette, add 2 drops of secondary antibody to all the wells you labeled in step 1. Add 2 drops of the substrate to each of the wells. Look for an immediate color change, indicating the presence of antibodies. These color changes must be observed within 2 minutes of the addition of this substrate. Record your results and discard your pipette. Students learn about Lyme disease, its cause, and how to detect the infection based on the ELISA test. ELISA tests are also used for the diagnosis of other infectious diseases, such as bird flu. Students learn about the immune system and experiment with an ELISA simulation test for detecting circulating antibodies in simulated patient serum samples. The first set of 2 wells are the negative control, with no color change. The second set of 2 wells are the positive control, with a color change. Patient samples showed a negative result for patient 1 and a positive result for patient 2. The result indicates that patient 2 is infected with the bacterium that causes Lyme disease.